A very good morning to one and all. I myself Pradnya khapekar from Jagdamba Mahavidyalaya Achalpur, and I am presenting a paper on the topic pharmacognostic approach and response of Artemisia pellens wall to WAM and alvar inoculations by root trainer technique. And firstly, we will see the introductory part. Now, in introduction, this Artemisia pallens is an aromatic annual herb, and it is used in Indian folk medicine for the treatment of diabetes mellitus. And this plant is also accredited with anthelmintic, antipyretic, and tonic properties. The oil which is extracted from this plant is antispasmodic and stimulant. So this was firstly proved by the Achalpur et al. In 1981, and it also possesses antibacterial and antifungal properties. Uh, the oil which is extracted, it is also is a high grade perfume oil, and also it is used as a flavoring agent in food items, beverages, and tobacco products. Now, the common name of this Artemisia pallens is Davna. It is also called as a Davna, and the oil which is extracted from this plant is also called as a Davna oil. So, this Davna oil is soothing to rough, dry, and the chapped skin. Infections which are seen on the skin and the cuts lesions are seen on the skin are also cured by this the Davna oil. Now, its unique and pleasant fragrance and attributes, they are, it has a large property like mucolytic, aphrodisiac, nerving, calmatives, emollient, and anti-infections. Now, the centonin is one of the product which is extracted from this oil, and this centonin is extracted from the flower heads present in the plant. Now, heads of the flower, it consists of a small the pollen grains and all. So from this the centonin is extracted, and it contains the volatile oil. Now this volatile oil, it also has a maximum action on the roundworms which are seen on the skin, and it was proved by the Kadri in 2010. Now the endomycorrhizal fungi, we will talk about the endomycorrhizal fungi, because the work which I am presenting in this paper is related to the mycorrhizosphere and the rhizosphere, which is the main part of the root, because root region is always in contact with the rhizospheric soil which is present all over the root. So this endomycorrhizal fungi, it first produce the branch hyphal structure within the plant cell. The root which are present, and surrounding the root is there is a rhizospheric soil. So these rhizospheric soil, it always consists of small we can say the granules of fungi or the hyphae of fungi which are present all over the root region. So this infection, it creates an absorptive structure with a very high surface area, where with the help of these endomycorrhizal fungi, there is a transfer of our protein which is a contact between the plant and the fungus. So it is a great channel which is present between the plant and the fungus, by which the proteins can be absorbed by these roots. Now, suppose we talk about the biofertilizers. The biofertilizers, they are commonly called as the microbial inoculants, which are capable of mobilizing the important nutritional elements in the soil from non-usable to usable form by the plant, which are through the biological process. Because these biofertilizers are the biofertilizers, means we can see that fertilizer which we use in the form of a biological agent, they are also called as a microbial inoculants, and these have the capacity for mobilizing the important nutritional elements which are used in the usable form to enable or to usable form by the plant through the biological process. Then we will talk about the materials and the method which are used for this analysis. Now, the culture of the glomus fasciculatum, they was obtained from the Tata Energy and the Resource Institute, New Delhi, and it was mass cultured under the grasses in open clay pots. The experiment was mainly it was conducted by the root trainer technique. Now, these root trainers are trays are filled up to the half of the capacity with the potting mixture pre-sterilized with methyl bromide. Mixture of that soil, it consists of 1 is to 1% of soil and the farmyard manure, which is in 2%. Goat compost was used, which was 0.25%. The rice husk was 0.25%. Charcoal granules were used, which was also in the constituent of 0.25. Uh, Single superphosphate was used, which was 0.25, and the VAM, the VAM which was particularly used was the glomus fasciculatum, which was used in the proportion of 2.5 to 3.5 gram per tube. Now seeds, they were firstly pre-soaked in water for overnight, and it was sown, and the saplings were allowed to grow in the root trainers for 1.5 months. Then the algal culture was prepared separately. This algal culture was inoculated in each tube, except the control, and 3 to 6 replicates were used for each treatment. And 100 tubes containing healthy saplings were selected, and these trays were kept in the open ground nursery site. The saplings were watered with tube well water, and cultured practices like weeding. All the weedings and were performed during this experiment was performed, and it was including all the it were protection measures were taken for the scorching sunlight and all, and the weeding process were done. Now seedlings from the root trainers, they were transferred to polybag after 1.5 months, and the effect of these biofertilizer, that is VAM and algae, they were seen simultaneously on the growth of medicinal plants, as which were conducted by the employing the root trainer technique. Then an inoculum which was consist of glomus spores, it was cultured in grasses in open clay pot for the multiplication of these spores of the glomus fasciculatum. While nostoc, they were cultured in the BG11 medium and was used to study the influence on the growth of medicinal plants. Then preliminary, the phytochemical investigation, it was shown for the presence of carbohydrates. Phytochemical investigation was done properly to study the plant in various minute effects. So what are the presence, which type of phytochemical products are present in that plant. So for that, various tests were performed, and these phytochemical is after the phytochemical investigation, it was seen from that test that there was the presence of carbohydrates, sugars, starch, protein, amino acids, glycogen, alkaloids, and tannins. Also, these carbohydrates, amino acid, and protein, they form the reserve food. Secondary metabolites such as glycosides, alkaloids, oil, tannins, and phenolics are responsible for the therapeutic use of this plant. Then the after all the conclusion, it was seen that after performing all the root trainers experiments and the polybag experiment, the results were noted. And first of all, the first table 1 shows the total dry matter production in Artemisia palens. In that, you can see that treatment were given. By the first treatment was of control, the second treatment was of only inoculation of glomus fasciculatum, and the third treatment was combination of glomus fasciculatum and the nostoc. Now the readings were taken by taking in account the height of the plant, fresh weight of the leaves, dry weight of the leaves, fresh weight of the root, fresh weight of the shoot, and all the treatment. Okay, so here you can see that increased result and the improved result was seen in the in the combination of glomus fasciculatum and the nostoc in comparison to the control and glomus fasciculatum. Here you can see the height of the plant is 74 centimeter in control, but in combination you can see the glomus fasciculatum and the nostoc combination shows the improved result in all of that. That is in the height of the plant, fresh weight of the leaves, dry weight of the leaves, fresh weight of the root, and fresh weight of the shoot. Then secondly, again the plant was studied by from the mycorrhizal point of view, and the chlorophyll content was also observed. So these in these the readings were taken by taking the first content, that is percent of VAM colonization, percent of vesicles, percent of arbuscules, and the chlorophyll content. Here also the improved result was seen in combination of glomus fasciculatum and the nostoc. Here you can see all the tables here. So all these shows the improved result, means percent of colonization is also more in combination of glomus fasciculum nostoc, percent of vesicles, percent of arbuscules, and also the chlorophyll content was also the maximum in that case. And lastly, uh, our data. Our data indicate the maximum and the improved results were observed due to the application of all mycorrhizal and cyanobacterial biofertilizer along with the combination of chemical fertilizer, small amount of chemical fertilizer. The plant showed improved growth in comparison to control. The control plant, they show the VAM associates varies and all the values were mean of the three replicates. These medicinal plants were first treated in root trainer and after 45 days they were transferred in the field where the final treatment was given and the results were observed. There was also increase in the biomass production, dry weight of root ensured due to the treatment of Glomus fasciculatum and Nostoc pallidusum. So these increased fresh and dry weight of leaf, they may also be due to the superiority of the treatment of with resulted in the vigorous vegetative growth parameters and yield attributes result in the higher values. Gandhi Kumar in 1996 he reported higher dry matter production in the Artemisia pelens with the application of azospirillum and nitrogen. So similar results were reported by the Sumechand et al. In 2001 and in the plant he worked in the menthol mint and all. That's all. So thank you.